My name is Marina Malakova, and I will be your instructor for today's webinar entitled Management of Phase 1 Clinical Trials. So a little bit about myself. I have over 18 years of experience in the clinical research field with about 11 years experience on the executive level. I have managed all phase clinical trials, phase 1, 2, 3, and 4 post-marketing studies involving different type of test agents, and mainly in the field of surgery, cancer diagnostics, and interventional radiology. In terms of my background, I am a scientist by training. I have PhD in biochemistry, and subsequently I moved to the States and completed my fellowships and attained also master's in clinical investigation. So in relevance to this webinar, what gives me a unique perspective is essentially understanding the science behind therapies, but also understanding the transition that needs to take place when you move from cell lines or animal models to first-in-human clinical trial. And subsequently, I got specific certification and regulatory aspects from the Society of Regulatory Affairs Professionals, RAC, and also focused on clinical safety and pharmacovigilance, and recently served as Vice President of North American Chapter for International Society of Pharmacovigilance. So safety is one of the latest aspects that I am educating myself. I also was privileged to edit and co-author a book, coincidentally published also by Barnett International, on biologics development, including transitions from preclinical to clinical phases, also elucidated in the book. And currently, in my daytime job, I serve as Executive Director of Research, managing all aspects of clinical trials, micromanagement of clinical research programs, including study design, financial aspects, quality, risk management, and strategic planning. Also, in relevance to its training series, I think I've been training with Barnett International close to 6 or 7 years by now, and for the past 14 years, I also have been an instructor at Boston University for courses such as Design and Conduct of Clinical Trials, Introductory Pharmacology, Clinical Trials Management, and Good Clinical Practices. So this is just in brief a little bit about my own background. Before we start, so what are the major sources of new drugs? The major sources of new drugs could be done by what's called organic synthesis. It could be new use of an old drug for the new indication, natural non-human chemicals, human chemical that's purified, and biologic that is, let's say, produced by gene therapy or some sort of like recombinant DNA technology is utilized. So those could be gene therapy, vaccines, proteins. So by its nature, those are macromolecules. So no matter how big and bulky chemical entity is, with like multiple functional groups, it's still a single molecule. So it's chemical entity consisting with traditional synthetic drug or purified drug. That's drug as a chemical entity. It's one molecule. Biologics are typically macromolecules. So they are a lot more complex, they are more sensitive to pressure, heat, and other external factors. So what questions you need to answer at the beginning of your planning? So when you embark on drug development, you need to decide what disease or condition do you want to treat? Where would you like to intervene in the disease process? What's the molecular target? What specific patient population will benefit? What are desired treatment outcomes? And what is the specific treatment administration in terms of dose and treatment duration? Is it acute condition that you're trying to alleviate? Is it chronic condition where extended exposure would be needed to alleviate, let's say, some sort of symptoms of that disease?